Hi guys, Michelle Glass here. Welcome back to another installment in our Chapter 17 lecture series. And our topic for this video and the following is on our sense of vision. Now, this is complicated since the anatomy is complicated, the physiology is complicated. I am actually just grazing the surface. When you really want to understand how our vision information is processed and a lot of how that information is coming into the eye. There is a lot more to this sensation than what I'm going to talk about in this video. There's a lot more in the textbook, so if you find this very interesting, I would recommend that you go ahead and read that section in your book and then bring your questions to me. Otherwise, pretty much the information presented in this presentation and the next will be, what I'll be testing over in a class. It gives you kind of just the basic foundations that you need, but just keep in mind, it's not, the whole picture. Did you like that pen? Of what's going on. Okay, so let us get started. So we need to first make sure we're comfortable with the anatomy of the eye. And so I have, I've made an attempt here to draw out for you the cut of the eyeball. Now you have been or will be studying this as part of your lab work, so you should already have a pretty good understanding, and if not, make sure that you're following along with my diagram on your notebook paper, and also take time to look at the diagrams in your text and the models in your lab and all of that. But to start with, we're going to see that the eye has three what are described as layers or tunics. So think about them as like layers in an onion, and we're going to start with the outermost layer and then move our way in. The very outermost part of the eyeball is described as the fibrous layer, and most of that is composed of the sclera. The sclera would be that hard white thick layer that's making the structure of the eyeball, and then also the cornea, which is just right over the front of the eyeball. The cornea is the transparent part that does allow light to enter. So light is not entering the sclera, but it can enter the cornea. As we move into the next layer or tunic, we are looking at the vascular layer or the vascular tunic. And this is composed primarily of the choroid. So the choroid is going to, as the name implies, the vascular layer is going to provide the vascularization to the eyeball. You also have at the front of the eyeball a circular muscle called the iris. The iris is controlling the diameter of the pupil. So the pupil, as you can see here, is just a hole. It's just an opening. Hopefully you get to do a dissection in your lab and you can really see that very well in the dissection. That's why I always like to do that. The iris then is a circular muscle like this, and so it's surrounding the opening of the eye. The pupil is the hole that's allowing the light to penetrate. Circular muscles, when they contract, that diameter gets smaller, and when they relax, that diameter gets larger. So to have a dilated pupil, you'd have a relaxed iris, and then to constrict the pupil, you would need to contract that muscle. The iris is the colored part of the eye, okay? Then you also have your lens. The lens is having the job of focusing that wavelength of light and holding the lens in place. You have the ciliary muscle, it has ciliary body, it has suspensory ligaments that are attached to the lens and holding the lens in place. Now, not only is the ciliary muscle sort of holding the lens there, but it also can control the shape of the lens and be important then in focusing the wavelengths of light. The ciliary muscle we'll look at a little bit more in a video just on what's called accommodation, which is really looking at the job of the ciliary muscle. But it is also, just keep in mind, it's also a circular muscle. So again, when it is contracted, it's a smaller diameter, and when it is relaxed, it is a larger diameter. And so these would all be components of the vascular tunic. If we go on the before we go to the next layer, you do have what's described as the anterior chamber and then the posterior chamber of the eye. The anterior chamber, the front part, would pretty much be inside that cornea to about the lens, and that is full of a fluid called aqueous humor, which is a very watery-based fluid that's really there to provide transportation for nutrients and oxygen and waste disposal to the cells there of the lens, especially, and the cornea, those transparent tissues where you don't want to have a lot of vascular tissue. Okay. And then the innermost layer of the eyeball is the neural layer or neural tunic, and this is composed of the retina. The retina contains your photoreceptors, which are going to be the cells that are stimulated by light or stimulated by photons. And then also we're going to see some neurons here as part of our retina. We're going to be looking at the retina, the cells of the retina here in just a few minutes. So we will see it's only a few cells thick. It's maybe 1, 2, about 4 cells thick. So it's a very, very thin layer. Now, holding the retina in place, keeping it at the back of the eyeball, and giving the structure to that whole eyeball itself is the vitreous humor. So the vitreous humor is much more viscous. Viscous here means very thick. And so, again, one of its key jobs is keeping that retina in place. And then also coming off the back of the eye, we have our optic nerve, which is our cranial nerve number 2. And this is the nerve that's taking the sensory information from the eye to the brain for processing. Okay, so let's take a closer look at that retina. Well, I jumped ahead. Okay, so really we're trying to trace how the light enters the eye, and then as part of that, we'll be looking at our retina in a little bit closer detail. So your particles of light are called photons, and the photon, before it actually enters the eyeball, is going to enter in another transparent tissue called the conjunctiva. So you have conjunctiva is the layer of tissue that's aligning the eyelid and then becoming continuous, going over top of the eyeball and then lining the inside of the lower lid. So it's going underneath the upper lid, over top of the eyeball, and then underneath the lower lid. When I was a kid, especially riding home on the school bus, I remember lots of the little kids like to flip up their eyelids, and it looks so gross to me, and it always made me squeal really loud. And that tissue that they were showing me was their conjunctiva. The palpebral conjunctiva is the conjunctiva that's underneath the eyelid. The ocular conjunctiva is the layer that's going over top of the eyeball, and it is transparent, so of course light can penetrate. So that's actually going to be our first layer here. And I haven't drawn that into the diagram. Okay, so next we would actually enter the cornea, pass through the aqueous humor. We're going through the pupil there, which remember is the opening that's controlled by the iris. And then we're going to go through the lens. Remember, it's the job of the lens to actually focus that light to our retina. So in order to get to the retinas, we have to go through that vitreous humor, and we go all the way to the back of the eyeball. Now, if you're looking directly at something, that wavelength of light should be focused directly at what's called the fovea centralis. This is a very specific part of your retina. The retina, remember, contains your photoreceptors, those cells that are stimulated by photons, and you actually have 2 types. You have what are called rod cells and cone cells. The fovea centralis is your point of highest or most accurate vision because this is your highest concentration of cone cells. The cone cells are the cells that allow you to detect color, and color vision is giving you the most accurate vision. So if you're looking directly at something and your lens is working correctly and your ciliary muscles are working correctly and all that, then you're going to get that wavelength of light focused directly back onto that fovea centralis, which again is part of the retina. So let's take a look. Now we're ready. Here we go to look at that retina. So we are looking at the very back of the retina. So right here, the pigmented epithelium is closest to that choroid layer. And the job of the pigmented epithelium is really just to absorb extra photons of light. The very next layer you have are the photoreceptors, and you have just one layer of these, but you do have both rod cells and cone cells. Now, if we were looking here at the fovea centralis, you would see primarily cone cells. Those rod cells are more on the periphery of the retina, and so it's giving you more of that peripheral vision. If you really want to figure out what's going on in your peripheral vision, you turn your head, and now you're getting that wavelength of light focused directly onto your fovea centralis. The photoreceptor cells synapse onto a neuron called a bipolar neuron, and it's named in part because, that's the shape of the cells here. And those bipolar neurons are synapsing onto your sensory neurons, which can also be called your ganglion neurons. Now, these neurons are going to be forming the optic nerve, which we've already said is cranial nerve number 2. So remember I said the retina is very thin. It's 4 layers of cells, pigmented epithelium at the back, so the wavelength of light is going all the way to the pigmented epithelium, coming back to the photoreceptor, and then the photoreceptor is stimulating the bipolar neurons, which are then sending that signal onto the sensory neurons. Now, even though there's just 4 layers of cells here, these are not the only cells that make up the retina. There are 2 additional types of neurons. So we have our horizontal cells, which are going to be in between the photoreceptors and the bipolar neurons. And then we have the amacrine cells, which are in between the bipolar neurons and the sensory neuron. And both of these cells have the job of sort of altering the retina's sensitivity and modifying the signal as it's sent. When we talk about a photoreceptor cell, it's stimulated by photons, wavelengths of light, particles of light. And in order to be able to be stimulated by light, you need to have a pigment. A pigment is a chemical that can absorb those photons. And in the case of rod cells, the pigment we have is rhodopsin. Now, different pigments have a different color to them because they're absorbing differently on that spectrum of light. So the reason that your rod cells are doing black and white vision is because they have the pigment rhodopsin. And then your cone cells can see different wavelengths of light because they have different pigment molecules. The rhodopsin is a combination of 2 components, opsin and then what's called cis-retinol. And notice here I've kind of drawn my retinol with a circle and then a little hook. So notice the hook. The term cis here means bent. So this is just chemist lingo. Cis here is referring to it being a bent shape, okay? So when we talk about a rod cell or a photoreceptor actually being stimulated by a photon, what we see happening is that the wavelengths of light, the photons, are absorbed by the rhodopsin, and during that absorption, it's going to convert your cis-retinol to what's called a trans-retinol. So it's going from a bent shape to a straight shape, okay? When that happens, when your retinol is straight, it's activating the opsin, and when opsin is activated, it's going to open up sodium channels. So we're going to get sodium influx. So now our rod cell is depolarized, and that action potential is going to be sent on to the bipolar neuron. Okay, so this is our basic mechanism. The rhodopsin absorbs photons, converts your bent retinol to a straight retinol, activating opsin, leading to sodium, moving into the cell, depolarization, action potential. Okay, but that's not the end of the story, because what we see is that that trans retinol has to, in some way, get converted back to the bent shape so that it can go back to, being activated again. And this takes several steps, so that's what we're going to be looking at next. Now, this process of taking a straight retinol back to a bent retinol is described as bleaching, because during this period of time, you're not able to stimulate that photon again. So this is in part why, if you have a light shined in your face and then the light's taken away and it's kind of black for a little bit before you're sort of blinded before you can see again, is because you have, activated a lot of these rod cells, and so they are bleached out. They're not able to be stimulated again for a little period of time. So in order to convert your transretinal back to cisretinal, you have to actually separate the opsin and the straight retinal. So here's my little just quick abbreviation for my transretinal. And then you're going to see ATP and enzymes are required to bend that retinal back to its cis shape. But this whole time, that opsin is bleached. There's no retinol there. It can't be stimulated again. It can't absorb any more photons. And so then it's after the retinol is back to its bent or cis shape that it can rejoin opsin and it's ready to be stimulated again. Okay, so let's see where we are. We have had our photons enter ocular conjunctiva, cornea, aqueous humor, pupil, lens, vitreous humor, or to the fovea centralis at the retina. Those photons, remember, go all the way to the back of the retina, so to that pigmented epithelium, and then they're going to be absorbed by the photoreceptors. The signal will be sent to the bipolar neuron, to the sensory neuron, which then becomes part of your optic nerve, nerve number 2. And then that information is going to be processed at the visual cortex, which is located in the occipital lobe. And that is, most of the physiology of the eye that we'll be covering in this chapter, although there's one little piece in the next video, so stay tuned.